Okay, good afternoon. We will continue with the discussion on the cerebral hemisphere, and now we will be on dealing with the internal structure of the cerebral hemisphere. This will consist of four areas: the lateral ventricle, the masses of gray matter, the basal nuclei, and the nerve fiber. So the lateral ventricle is paired. You can see that um, in both hemispheres. So one in a cerebral hemisphere. This is C-shaped cavity lined with ependyma and contain your cerebrospinal fluid. And if you remember, the lateral ventricle is the largest ventricle containing your CSF or producing this is the ventricle which produces the most CSF or cerebrospinal fluid. So it has a body and horns. The body will be found in the parietal lobe, and it will extend from the interventricular foramen where the CSF will flow to the third ventricle and to the posterior end of the thalamus. So that will be the body. And the body will have a roof which will be made up of the corpus callosum. Remember the white matter in the medial surface of the cerebral hemisphere. And it has a floor formed by the body of the caudate nucleus and the lateral margin of the thalamus. Its medial wall will be made up by the fusion of the roof and the floor, and also by your septum pellucidum. Now, the most, there will be horns in the lateral ventricle, and the most anterior horn or the frontal horn or the one in the frontal area will be your anterior horn. This will extend from the parietal lobe to the frontal lobe. And this will consist of a roof which will be formed by the anterior part of the corpus callosum, and the floor will be made up by the head of the caudate nucleus. The medial wall will be formed by the septum pellucidum and the anterior column of fornix. Now, the posterior horn will extend from the parietal lobe to the occipital lobe. So it will go posterior. And it will consist of a roof and a lateral wall made up of the tapetum of the corpus callosum and the optic radiation. The medial wall will be formed by the forceps major or the bulb of the posterior horn, and also made up by the calcar abyss. So there will be two structures that will form the medial wall of the posterior horn. You have the forceps major and the calcar abyss. Then the inferior horn will be from they will extend from the parietal lobe to the temporal lobe. So it will be running. You have to remember that the body of the lateral ventricle is found in the parietal lobe, and these horns will be projections to the different lobes of the cerebral hemisphere. So your inferior horn will be projecting into your from your parietal lobe to your temporal lobe. So it will consist of the roof formed by the tapetum of the corpus callosum, just like your anterior horn, and the tail of the caudate nucleus. And the floor will be formed by the collateral eminence laterally and the hippocampus medially. So the significant terms that you will encounter in the study of the internal structure of the cerebral hemisphere, you have the amygdaloid nucleus. You have encountered this already in the limbic, in the lecture on the limbic system. It is the anterior end of the tail of the caudate nucleus. Then you have the stria terminalis, which is medial to the tail of the caudate nucleus and ends in the amygdaloid nucleus. You have also the pes hippocampus, which is slightly furrowed anteriorly expanded end of the hippocampus. And then you have the alveus, which is a thin layer of white matter covering the ventricular surface of the hippocampus. Then you have the fimbria, which are bundles of axon of the hippocampal cells in the medial side of the hippocampus. Then you have the optic radiation, which are nerve fibers posterior to the posterior limb of the internal capsule, radiating to the calcarine sulcus. So you have another structure is your basal nuclei. These are also known as your basal ganglia, which are collection of masses of gray matter in the cerebral hemisphere. This will consist of the corpus striatum found lateral to the thalamus, divided by the internal capsule into caudate nucleus medially and the lentiform nucleus laterally. So your caudate nucleus is a C-shaped mass of gray matter, which is laterally, you will have the internal capsule, and medially, it will be related to the thalamus. The lentiform nucleus is a wedge-shaped mass of gray matter, which is broad convex base, directed laterally, with the blade medially directed. Then laterally, your dentiform capsule is related to your external capsule, separating it from the claustrum. While medially, it will be related to your internal capsule, separating it from the thalamus and the caudate nucleus. Then you have the functions of the corpus striatum, which is to control muscular movement through the cerebral cortex rather than through the descending pathway to the brainstem and the spinal cord. So the corpus striatum will have a faster control of muscular movement compared to the pathway, the descending pathway in the spinal cord. Then you have the amygdaloid nucleus found in the temporal lobe close to the uncus, which is part of the limbic system. Then you have the claustrum, which is a thin sheet of gray matter with a known function. Laterally, it is related to your subcortical white matter of the insula and medially to the external capsule. Then you have the septum pellucidum. This is a thin vertical sheet of nervous tissue made of white and gray matter covered by ependyma. This is located between the fornix and the corpus callosum. Anteriorly, it will occupy the interval between the body and rostrum of the corpus callosum and forms a portion between the anterior horn of the lateral ventricle. Then you have the thelacoroidea. This is a two-layered fold of pia mater forming the choroid plexus of the ventricle. It will extend from the fornix to the roof of the third ventricle, or the upper surface of the thalami, and covered by ependymal when projecting into the ventricles. Your thelacoroidea has an arterial supply coming from the choroidal branches of your internal carotid artery and your basilar artery, while its drainage will be from the internal cerebral vein. It will drain into the great cerebral vein to the inferior sagittal sinus and finally to the straight sinus. Structural and functional localization of the cerebral cortex. So your cerebral cortex is a gray matter covering of your cerebral hemisphere, which is thickest at the crest of the gyrus or the upper portion of the gyrus and thinnest at the depth of the sulci. So it will consist of nerve cells, nerve fibers, neuroglia, and blood vessels. So there are 6 types of nerve cells. You have the pyramidal cells. That's why it is called pyramidal cells, because of its shape, which is about 10 to 15 angstrom in size. And you have the biggest pyramidal cells, which are your Bett cells. They are in the motor precentral gyrus of the frontal lobe, measuring 120 angstrom. So it is how many times is it doubled than your pyramidal cells, the common pyramidal cells. Then you have the third type is your stellate cell or your granule cells. These are small and polygonal cells measuring 8 angstrom in diameter with multiple branching dendrite and short axon terminating on a nearby neuron. Then you have the fusiform cell, which are small cells oriented vertically along the long axis of the surface, located in the deepest cortical layer. The axon of your fusiform cell will arise in the inferior part of the cell going into the white matter as projection, association, or commissural fibers. So your fusiform cell will be traveling outside of the gray matter to go into the white matter. And this will form specific fibers. Now, the dendrites will arise from both poles, wherein the superficial dendrites will ascend and branch in the superficial layer, while the inferior dendrite will branch out in the same layer. So, your fusiform cell will have several dendrites that will come out from both poles. Then the horizontal cells of Cajal. These are small fusiform cells oriented horizontal to the cerebral hemisphere. And the axon will connect with the dendrite of the pyramidal cells. The dendrites will arise from both poles of cell body. So your horizontal cell of Cajal, just like your fusiform cell, will have many dendrites. Then you have the cells of Martinotti. These are small multipolar cells found in all levels of the cortex. Dendrites are short, while axons are directed to the pial surface of the cortex, giving short collateral branches. So meaning to say, your cells of Martinotti, the axons will not go to the white matter, but will remain in the gray matter, and it will go to the pial surface. So there, you have also the nerve fibers. There are 2 types. You have the radial fibers, which are oriented at right angle to the cortical surface and made of afferents coming from projection, commissural, and association fibers, and axons of pyramidal, stellate, and fusiform cell, which end in the white matter. So your radial fibers can be said that it is in the white matter. The tangential fibers, this is oriented parallel to the cerebral cortex. Is made up of collateral and terminal branches of afferent fibers and axons of horizontal and stellate cells and collateral branches of fusiform and pyramidal cells. So this is the picture of the cells and fibers in your cerebral cortex. So you have here the basket cells. Then you have the fusiform cells. You have here the horizontal cells of Cajal. There. You have to take note that the horizontal cells, it stays on the first layer of the cortex. Then you have the cells of Martinotti. Then you have the neuroglial form, the pyramidal cells. The pyramidal cells is well distributed on the different layers of the cerebral cortex and the stellate cell. So, here. So you can notice that the pyramidal cell is well distributed, while your horizontal cells of Gall prefers to stay in the first layer of the cortex. So there are 6 layers also of the cerebral cortex. So when you say that when cerebral cortex in the area of the cerebral cortex has these 6 layers, they are called as homotypical. So the first layer or outermost layer is the molecular or plexiform layer. So this is the first layer. So that is your molecular or plexiform layer. And here there will be most synapse of the neurons could be found. So the derivatives of the nerve fibers that you will see in this layer, you will have the apical dendrites of the pyramidal and fusiform cell, the axons of stellate and Martinotti cells, the afferent fibers from the thalamus associated with commissural fibers, and the horizontal cells of Cajal. Then the next layer, going more deeper, you have the external granular layer, which consists of abundant small pyramidal and stellate cell whose axon ascend to the molecular layer and the dendrite descend to the white matter of the cerebral hemisphere. Then the external pyramidal layer, this will consist of pyramidal cells with upgrading cell body sizes as it goes deeper. So the external pyramidal layer will have many pyramidal cells, and as the cell goes down or become deeper, it becomes bigger. So probably we can see the best cell, most of the best cell here. The apical dendrites pass to the molecular, so first layer, and axons enter the white matter as projection, commissural, and association fibers. So this will be the content of your the axons will go to the white matter, while the apical dendrites will go to the first layer, to the molecular layer. Then the fourth layer is the internal granular layer, composed of closely packed stellate cell and external band of bilarger. And the fifth layer is the internal pyramidal or ganglionic layer, consisting of large and medium-sized pyramidal cells interspersed with stellate cells of Martinotti and inner band of bilarger. I want you to research what is this band, external and internal band of bilarger, and you give your answer by sending to my Facebook page, Facebook account. So that's the first question. What is the, or differentiate, what is the inner and external band of bilarger? Multiform layer or layer of polymorphic cells are cells which consist of abundant fusiform cell, modified pyramidal cells, and cells of Martinotti, and nerve fiber leaving or entering the white matter. So you have to remember the multiform layer is the innermost layer. So the most of the nerve fiber will enter the white matter because this is now nearer the white matter. Then those 6 layers, as I have said earlier, if it is found in a certain area of the cortex, you can say that the layers of the cerebral cortex is homotypical. But it is not this in the same way in all areas of the cortex. There will be some cortical layer which will not possess the 6 layers. So they are called heterotypical type of cortical layer. The reason why they do not possess the 6 layer because there will be fusion of layers. So here, the types of heterotypical type of cortical layer, you have the granular type. This contains densely packed stellate cells. Here, there will be a fusion of your layer 2 to 4. So that will be your layer 2 to 4. So it's fusion of your external granular layer, your external pyramidal layer, and your internal granular layer. So that is the granular type of cortical layer. Here it will be predominantly of the granular cells which receives thalamocortical fibers. You can see this granular type of cortical layer in the post-central gyrus. So it will be from post-central gyrus, it will be somatosensory. And the superior temporal gyrus and the hippocampal part of the hippocampal gyrus. The granular type will have a well-developed layer 3 and 5. So it would be layer 3 and 5. It will be your external pyramidal layer and your internal pyramidal layer, which will be very much developed if it is of the granular type. Now it will be made up of pyramidal cells of various sizes, and the efferent fiber will be associated with motor function. The location is in the precentral gyrus and other areas of the frontal lobe. Because remember, your agranular type, you can find this in motor area. Precentral siya. So the cortical area based on anatomical location. The frontal lobe, you have the precentral area in the precentral gyrus. Histologically made up of pyramidal cells, and it is divided into portions. You have the posterior portion is your motor area or primary motor area or the Brodmann's area 4. So this is in the frontal lobe. And it will extend from the precentral gyrus to the paracentral lobule. So from the lateral surface, you remember that your precentral gyrus is in the lateral surface of the cerebral hemisphere. It will fly over the surface of the cerebral hemisphere to go to the medial surface where your paracentral lobule is located. Then the anterior portion is the premotor area. This is also known as the secondary motor area, or Brodmann's, sorry for that, there should only be one N here. Brodmann's area number 6 and part of area 8, 44, and 45. So these are the names of the anterior portion of the anterior portion of the precentral area of the frontal lobe. Now it is wider, the anterior portion is wider superiorly, and it will taper inferiorly and be confined in the anterior part of the precentral gyrus. So there is no giant pyramidal bed cells here in the anterior portion. The function is the primary motor area is for the execution of individual movement of the different parts of the body through the afferent fibers from the premotor area, sensory cortex, the thalamus, cerebellum, and the basal ganglia. So meaning to say, in the premotor area, the execution of the movement will be aided also coming from afferent fibers that will come from the premotor area, the sensory cortex, the thalamus, the cerebellum, and the basal ganglia. So although the preconus area is the motor area, it will be aided. Afferent fibers will also aid in coming from the different areas like your cerebellum, your basal ganglia, and your thalamus. So your premotor area store programs of motor activity assembled as a result of past experience. Thus, it will regulate the activity of the primary motor area, especially coarse postural movement through its connection with the basal ganglia, sensory cortex, and the thalamus. So your premotor area is more of for storage of the movement that has been learned or experienced before. So when can you use this premotor area? When you are joining in a dance competition. So you have to store all those movements in your premotor area so that in the actual dance performance you can get the signal from your premotor area. So movement areas of the body is represented in an inverted form in the precentral gyrus and positioned in such a way that it coincides with the developmental stage of the being. This is known as your motor homunculus or precentral gyrus, which I will let you see in the next slide. So motor development in human beings progressively follow from inferolateral to superomedial organs and anatomic structure as mentioned in the illustration in your motor homunculus, which is this. So this will follow also, the development here will follow also with our growth. Remember, a baby can use first the tongue and the pharynx because how can it feed itself? So the baby or the developing neonate can first recognize the smell of the caregiver even before he or she can see, because the eye development is much later. But the baby can, by the smell, can recognize the caregiver. And he can reach things first before he can walk. So that is the meaning of your homunculus illustration. So other areas here in the cortex, on the frontal cortex, is your supplementary motor area. This is located in the medial frontal gyrus anterior to the paracentral lobule on the medial surface of the cerebrum. So your supplementary motor area is found on the medial surface of the cerebrum. And it will stimulation of this area will cause contralateral movement of limbs with the stimulus stronger than that needed by the primary motor area. There is no permanent loss of motor movement with the removal of supplementary motor area, because it is just a supplement, or it will just help with the movement. So much stronger good and primary motor area. Then the frontal eye field. This is located on the facial area of the precentral gyrus to the middle frontal gyrus, or part of your Brodmann's area 6, 8, 9. The function is, if it will be stimulated, it will lead to conjugate movement of the eye, especially towards the opposite side. Controlled voluntary scanning movement of the eyes, which is independent of visual stimuli. Then you have the motor speech area of Brocka. This is found in the inferior frontal gyrus, between the anterior and ascending posterior rami of the lateral fissure. Remember that your inferior frontal gyrus will be traversed by your lateral fissure of sylvius. Now, it will stimulation of this area, in the inferior frontal gyrus. It will bring about formation of words in conjunction with stimulation of the primary motor area, innervating the muscles of the larynx, mouth, tongue, soft palate, and respiratory muscles. So this will be between the stimulation of when you talk. Ablation of this area is in non-dominant hemisphere, has no effect on speech, contrary to ablation on the dominant hemisphere. Then you have the prefrontal cortex. This is located anterior to the precentral area, encompassing the greater part of the frontal gyri, orbital gyri, and the anterior half of the cingulate gyrus. So this is your Brodmann's area 9, 10, 11, 12. Now, this will function as a regulator of the person's depth of feeling and exert influence in determining the initiative judgment of an individual through its many connection with other areas of the cortex, like your thalamus, hypothalamus, corpus striatum, and your cerebellum. Now, the parietal lobe has three somesthetic area. Remember, your parietal lobe is the sensory lobe. And this is located posterior to the post-central gyrus. Your primary somesthetic area is found in the post-central gyrus on the lateral surface of the hemisphere and the posterior part of the paracentral lobule on the medial surface. This is your Brodmann's area 312. And the function will be for extensive sensory function on both contralateral and ipsilateral side. The secondary somesthetic area is found in the superior lip of the posterior limb of the lateral fissure. It has an unknown function. If you are interested to be a neuroanatomist, you may find this as a good research. Another area is the somesthetic association area. This occupies the superior parietal lobule extending to the medial surface of the hemisphere. So from the superior parietal lobule, it will jump to the medial surface of the hemisphere, and you will still find this somesthetic association area. And this will receive and integrate the different sensory modalities. The occipital lobe is the visual lobe. It has three areas for visual function. The primary visual area is the Brodmann's area 17, which is located on the walls of the posterior part of the calcarine sulcus and sometimes extend around the occipital pole onto the lateral surface of the hemisphere. Now, the function is that you will have receptors of visual stimulus coming from specific area of the retina. Visual cortex receives fibers from the temporal half of the ipsilateral retina and nasal half of the contralateral retina. Right half of field of vision is represented in the visual cortex of the cerebral hemisphere. You have the macula lutea represented by the posterior part of Brodmann's area 17 and the periphery by your ora serrata by the anterior part of Brodmann's area 17. The cerebral visual pathway, visual afferent fiber passing through the white matter of the temporal lobe will go to the lateral geniculate body. This is found in your thalamus, in the posterior portion. And it will go to your visual cortex, which is your occipital lobe. The secondary visual area surrounds the primary visual area on the medial and lateral surface of the hemisphere. And this will relate visual information received by the primary visual area to past visual experience. So if it is secondary, you can say that it is more for storage of past experience. Occipital eye field. This is probably located in the secondary visual area in man, and it will stimulation of the occipital eye field will lead to reflex movement of the eyes when following a moving object. Now, the temporal lobe is the auditory lobe. So, for your hearing, it has three areas. The primary auditory area is also known as your Brodmann's area 41 and 42. Now, the location is in the inferior wall of the lateral sulcus, including the gyrus of Heschel. It will receive sound, in which the anterior part of your primary auditory area will receive low-frequency sound, while the posterior part will be for high-frequency sound. You have also the cortical auditory pathway from the, so for audition or for it will be on the medial geniculate body. This is still a nucleus found in the thalamus. Projection fiber will go to the auditory area, then it will be received by the auditory radiation of the internal capsule. The second auditory area, or your auditory associated cortex, or your Brodmann's area 22, is found in the posterior to the primary auditory area in the lateral sulcus and the superior temporal gyrus. This is for interpretation of sound, your secondary auditory area. Then you have the sensory speech area of Wernicke's. This is in the superior temporal gyrus of the left dominant hemisphere and may extend to the posterior end of the lateral sulcus into the parietal region. This will allow the understanding of the written and spoken language and enable a person to read and understand and say out loud a sentence. Now, the cerebral cortex has an inner white matter. Here, neuroglia and the myelinated nerve fiber of the cerebral hemisphere will be located. Remember earlier when we talked about the cells, their fibers will go, some, except for your horizontal cells of Cajal, those fibers may go into the white matter, especially on the sixth layer. The classification, there will be commissural fibers, association fibers, projection fibers that will be seen in the white matter of the cerebral cortex. Their nerve cells will be seen in the gray matter. So the commissures, these will connect corresponding regions of the two hemispheres. The biggest and largest is the corpus callosum. Remember, the white matter in the medial surface of the cerebral hemisphere, which is a sort of curve C on the medial surface, middle portion. This will connect the cerebral hemisphere because the fibers of this corpus callosum will be projecting to the other cerebral hemisphere. Please take note of this. The corpus callosum, if you will, in the practical exam, sometimes Dr. Gasco will sort of separate the fibers of your corpus callosum, and he will make you identify what it is. If you know the location of the corpus callosum and you see a white matter on the medial surface, that you will know that that is the corpus callosum, because you might not recognize it because the fibers has not been separated. Now, this is located at the bottom of the longitudinal fissure. So this has several parts. You have the thin anterior end, which is known as your rostrum. This remember that the corpus callosum is sort of an inverted C. So the anterior portion, which is the thin, is known as your rostrum. And it will continue posteriorly with the lamina terminalis. Then the curved anterior anterior part of your, anterior to the rostrum is the genu. That's why this is called a genu, because in Latin it is termed as knee or bended knee. It is a curved anterior and bending inferior in front of the septum pellucidum. Then the greater part of the corpus callosum is the body. It will arch posteriorly and will thicken. Then at the posterior end will be uh, the, the splenium. This is the thickened posterior end of the body of the corpus callosum. Now, there will be fiber specialization. You have the forceps minor, which are fibers of the genu curving anteriorly to the frontal lobe. This is what I mean by the fibers that will be made by Dr. Gasco to be sort of the picture is like it's an unruly hair coming from the corpus callosum. Then you have the radiation of the corpus callosum, which are lateral extension of the fiber of the body. Then you have the tapetum, the fibers that participate in the formation of the roof and lateral wall of the inferior horn. Then you have the forceps major, the fibers arching posteriorly to the occipital lobe. Then you have the anterior commissure, small bundle of nerve fiber which cross the midline at the level of the lamina terminalis. It will extend anteriorly to the anterior perforated substance on the olfactory tract and posteriorly to the temporal lobe. Then you have another commissure, the posterior commissure. This is a bundle of fiber that cross the midline immediately above the opening of the cerebral aqueduct into the third ventricle. So you can see that these commissures will cross. It will cross. The nerve fibers from in the posterior commissure, you will have nerve fibers that from the pretectal nuclei, which will be involved in pupillary light reflex and is said to pass in this commissure on its way to the parasympathetic part of the oculomotor nuclei. Then another commissure is your habenular commissure. This is just a small bundle of nerve fibers that cross the midline above the root of the pineal stalk. So this is the habenular commissure is more seen on the inferior portion. This is associated with habenular nuclei whose afferent fibers comes from the amygdaloid nuclei and the hippocampus through your stria medullaris thalami. The function is unknown. Then you have the fornix, which are myelinated fibers making up the afferent system of the hippocampus and pass to the mammillary bodies of the hypothalamus. So the formation of your fornix, you have your nerve fibers that will form, which are thin layer of white matter covering the hippocampus. This is known as your alveus. So this layer of thin layer of white matter will converge and it will thicken to form now your fimbria. And it will go posterior to arch forward above the thalamus and below the corpus thalamus. And this will now be, as the fibria further thickens, it will now form your posterior column of fornix. Now, these posterior columns of fornix will converge at the midline together with the commissure of the fornix to form now your body of the fornix. This will connect the hippocampal formation of the two sides. Then you have association. From your commissures, you have your association fibers. These are nerve fibers that connect different cortical areas on the same side or hemisphere. So there will be two types, the short association fiber and the long association fibers. You have the uncinate fasciculus, which connect the first motor speech area and gyri on the inferior surface of the frontal lobe with the cortex of the whole of the temporal lobe. Then you have the cingulum, which is the long curved fasciculus lying within the white matter of the cingulate gyrus, and connection will be between frontal and parietal lobe with parahippocampal and adjacent temporal region. So this will be more of connection in the medial surface because that's where your perohippocampal and parahippocampal is seen. Then you have the superior longitudinal fasciculus, which connects anterior part of the frontal lobe with occipital and temporal lobe. You have another connection, your inferior longitudinal fasciculus, which will be seen from the occipital lobe passing lateral to the optic radiation to the temporal lobe. Then you have the fronto-occipital fasciculus, which connects the frontal lobe with occipital and temporal lobe within the cerebral hemisphere, lateral to the caudate nucleus. Then you have another, the projection fibers. You have afferent and efferent nerve fibers traveling from brainstem to all parts of the cerebral cortex, passing through large nuclear masses in the gray matter. This lies medial to the association fiber, intersecting the anterior commissure and the corpus callosum. This will form a compact band, the internal capsule, which will be bounded medially by your caudate nucleus and the thalamus, and laterally by the lentiform nucleus. So you will have an anterior limb and a posterior limb and a genu. Between the anterior limb and the posterior limb will be the genu. This will be radiating projection fibers emerging from the nuclear mass forming your corona radiata. Other cortical areas are your taste areas. This is located at the lower end of the postcentral gyrus in the superior wall of the lateral sulcus and adjacent areas of the insula. And then you have the vestibular area located in the postcentral gyrus, opposite the auditory area in the superior temporal gyrus. Then you have the insula. So you see that Snell consider only the insula as other cortical areas. This is located in the floor of the lateral sulcus, which is related to visceral function. Then you have association cortex. These are homotypical cortex, so therefore has 6 cellular layers. It has multiple inputs and outputs needed for determining behavior, discrimination, and interpretation of sensory experience. The recognized main association area and its function are your prefrontal cortex. This associate experience needed for the production of abstract ideas, judgment, emotional feelings, and personality. Then your anterior temporal will be for the storage of previous sensory experiences. Then you have the posterior parietal cortex. Here, it functions as an integration of sensory input of touch, pressure, and proprioception into concepts of size, form, and texture, appreciation of body image. So you have to remember that this main association areas are homotypical. So your posterior parietal, you have integration of input of touch, pressure, and proprioception into concepts of size, form, and texture, and appreciation of body image. Now, the cerebral hemisphere will have more or less equal function, more or less have a similar identity, but in terms of the gyri and sulci, it will be sort of identical. And the nervous pathway projecting contralaterally to the same cortical area and commissure, bridging the two hemispheres, providing equal inputs. But still, there is dominance that will prevail, performed by one hemisphere. Dominant hemisphere is concerned with handedness, perception of language and speech, while non-dominant hemisphere concerned with spatial perception, recognition of faces, and music. So meaning to say, when you are right-handed, your left left cerebral hemisphere is more dominant because remember it crosses. More descending fibers decussate at the pyramid. Speech area of an adult cortex is larger than the left. It's larger in the left than in the right. Transference of speech control is possible when lesion to this area occurs before the skill is learned. Right-handedness occurs in 90% of the population, and the remaining are left-handed and with some ambidextrous or dexterity in the use of left and right hand. Because you can still teach, like you can teach if you are right-handed and then you sort of have an accident affecting your right hand, you can teach your left hand to write. I remember I had a colleague who has a son who was about to take the entrance exam to Philippine Science High School, and weeks before that he had an accident during a basketball game which injured his right hand. So it would had a difficulty in writing with the left hand, but he taught himself and he was able to write with the left hand. While ambidextrosity means being able to use left and right hand with ease. So we go back to the cortical lesions and its manifestation. When you say expressive aphasia, there is loss of ability to produce speech, but the patient can think, write, and understand the word when seen or heard. So, going fast forward, if you are given a patient who has this condition, how can you extract the history? Simple. You just give a a pen and a paper to him, to the patient, and then you ask the question and you tell the patient to put the answer or to write the answer. That is for expressive aphasia, and it is an injury to the left inferior frontal gyrus affecting the motor speech area of Brocka. So that's why the patient cannot speak. Then for the next cortical lesion, which is your receptive aphasia, there is loss of ability to understand the spoken and written word and without impaired ability to speak. So the patient can talk and talk and talk. So the patient, when you will face him, given given a case, if you are given a case in this, what you will do is just listen to what he will be talking about. Because this patient, of course, will talk about his condition. So you just listen, and it will be you who will write what he has been talking about. So in receptive aphasia, the lesion is in the sensory speech area of Wernicke. Then we go to the third cortical lesion. This is a silent aphasia. Why do I say a silent? Because the patient cannot produce speech, it cannot understand spoken and written words. So what will you do so that you can extract an S history? You as the accompanying the the person accompanying the patient, because more or less he knows what happened and which leads to this lesion in the motor and sensory speech area. For alexianagraphia, here there is an ability to read or write. So in a case of a 1-year-old or the one who have not had the ability yet to read or write, of course they don't have agraphia, they don't have alexia, and there is no destructive lesion in the angular gyrus in the posterior parietal lobe. So what will you do to extract the history of this patient? Talk to him, because even if he cannot read or write, he has lost the ability to read or write, he can still speak and he can still understand you because he can still hear you. For a stereognosis, I want you to tell me by sending your answer to my Facebook account, what is the cortex that has been destroyed with a stereognosis, and how will you evaluate if the patient has this disorder. Okay, that's about, I think, all of my lecture, and I hope I have given you enough knowledge on the cerebral cortex, the external and internal anatomy of your cerebral hemisphere. Okay, good day and good night. J.